So, I want you to imagine an organism with just a totally wild identity crisis. Picture this, right? It is a perfectly normal, completely harmless resident of your own gut.
Right.
But then, it is also the exact same thing that public health officials look for to test if your local beach is safe for swimming.
Yeah, the water quality tests.
Exactly. And at the same time, it's literally a health supplement you can buy off the shelf at a local pharmacy. And yet, somehow, it is simultaneously one of the most terrifying, unkillable superbugs lurking in modern hospitals today.
I mean, it really sounds like an impossible riddle. You'd think we were talking about, four completely different things, but that entire resume belongs to just one single microscopic entity.
Well, welcome to this deep dive into the source material on Enterococcus, which is an organism I am officially dubbing the ultimate gut survivor.
I like that title. It fits perfectly.
Thanks. So our mission today is to unpack how this microscopic organism's sheer, stubborn hardiness drives absolutely everything about it. We're going to look at how it forced scientists to completely rewrite textbooks, how it alters global agricultural policies, and ultimately equipped you with a first principles understanding of a major healthcare threat.
And, a first principles approach is just vital here because trying to just memorize random facts about this organism without understanding the evolutionary logic behind, well, why it acts the way it does, that's just a recipe for confusion.
Yeah, I am still stuck on the paradox of it all, honestly. I mean, we are going to be talking about ward-clearing hospital pathogens today, which makes it totally baffling to me that I can walk into a health food store right now and buy a bottle of this stuff as a gut probiotic.
It's basically the ultimate microbiological paradox. And to understand how this organism pulls off all those different identities, we really first have to understand what it actually is, which, as it turns out, took scientists nearly a century to figure out.
Wow, a century. Okay, so let's start at the absolute basics. If you were to, say, put a swab of this under a microscope today, what are you actually looking at?
So you're looking at bacteria that are classified as gram-positive, catalase-negative, facultatively anaerobic cocci.
Okay, stop. We definitely need to translate that for everyone.
Fair enough.
So cocci means they are spherical, right? Like tiny microscopic balls, and usually they appear singly or in pairs or maybe in short chains. And facultatively anaerobic just means they can survive with or without oxygen.
Exactly.
But catalase negative, that is a term that gets thrown around a lot in biology, but break that down for someone who hasn't taken microbiology in like a decade.
Right, so catalase is an enzyme that a lot of living things use to break down hydrogen peroxide into water and oxygen.
Like when you pour hydrogen peroxide on a cut and it bubbles up.
Yes, exactly. That bubbling is catalase at work. But Enterococcus lacks this enzyme completely. It's catalase negative. So this tells us its internal metabolism is entirely different from a lot of other common bacteria, like Staphylococcus, which is catalase positive.
Oh, I see.
Yeah, it relies on entirely different biochemical pathways to survive.
So even at a fundamental metabolic level, it's just playing by its own rules.
It really is. And the history of naming it is a whole saga in itself. Back in 1899, there was this French physician named André Thiercelin. He isolated a gram-positive diplococcus, meaning those spheres that appeared in pairs from the human intestine.
Okay.
And he coined the term enterococcus, which literally translates to intestinal coccus. He just needed a way to distinguish it from all the other spherical bacteria scientists were cataloging at the time.
So Enterococcus literally just translates to gut sphere. That is brilliantly simple.
It is. But, well, the simplicity didn't last. For decades after Thiercelin, the organism just got completely lumped into the literature of a totally different genus, Streptococcus.
Wait, Streptococcus, as in the family that causes strep throat.
The very same. And this brings us to a really towering figure in microbiology, Rebecca Lansfield. In 1933, she developed this incredibly systematic way to classify Streptococci using serology.
Right.
She basically categorized them based on specific carbohydrate antigens that were present on their cell walls.
So she was basically sorting them by reading the molecular name tags stitched to the outside of their cells.
That is a fantastic way to visualize it. And those intestinal cocci that Thiercelin found, they possess the carbohydrate antigen that Lansfield designated as group D.
Ah.
So for the next 50 years, clinicians and microbiologists just referred to these organisms as Group D streptococci or fecal streptococci.
That's crazy.
Yeah, if you or anyone listening took a microbiology class literally any time before the mid-1980s, you were taught that this was just a quirky subgroup of streptococcus.
So for half a century, the entire medical world basically had it misfiled in the wrong cabinet. What finally broke the illusion?
The 1984 revolution. Two German microbiologists, Karl-Heinz Schleifer and René Kuiper-Balz, they decided to stop looking at those surface name tags and start looking at the actual genetic blueprints.
Okay.
They used a technique called DNA hybridization to compare what was then called Streptococcus faecalis to, well, true streptococci.
Wait, DNA hybridization. I think I remember this. Is that where they literally unzip the double helix of the DNA from both bacteria, mix them together, and just see if they stick to each other?
That is exactly how it works. You heat the DNA up to separate the two strands, mix the strands from organism A and organism B, and then cool them down.
Right, because of the base pairs?
Exactly. If the organisms are closely related, their genetic sequences will match up and zip back together tightly. But if they're distant cousins, the strands just sort of float past each other or they bond very weakly.
And when they mixed the gut sphere DNA with the strep throat DNA?
They realized they had a massive mismatch on their hands. The genetics didn't line up at all. The surface antigens had totally lied to them.
But I have to ask, why does this actually matter outside of an academic journal? Because taxonomy usually just sounds like academics arguing over flashcard vocabulary.
Right, it can seem that way.
Yeah, I mean, if a patient is lying in a hospital bed with an infection, does the doctor really care if we call it Group D strep or Enterococcus?
They should care, because taxonomy is actually a hypothesis about evolutionary relatedness. The Lancefield grouping, because it was based just on those surface antigens, it completely failed to capture how fundamentally different this organism's evolutionary path was.
Oh, I see.
By elevating Enterococcus to its own distinct genus in 1984, Schleifer and Kuiper-Balz were officially signaling to the medical community that, hey, this bug has a completely different survival toolkit than Streptococcus.
So it's kind of like realizing the creature rummaging through your trash isn't just a weird-looking house cat, it's actually a wolverine.
Yes, perfect.
You can't just try to shoo it away with a broom. You need an entirely different strategy because its biology is built for something else entirely.
And that Wolverine analogy leads us perfectly into the most crucial point of this entire discussion. The defining superpower of that unique evolutionary branch is just its extreme hardiness.
Hardiness?
Yeah, hardiness is the absolute organizing principle of Enterococcus. Everything clinically important about it flows from this one simple fact. It is built to survive hostile environments.
Okay, so let's talk about the main culprits in the human body then. You have Enterococcus faecalis, which historically causes about 80 to 90% of clinical infections, I believe.
That's right.
And then you have Enterococcus faecium, which makes up a smaller slice, but is rising super fast because it is incredibly good at acquiring antibiotic resistance.
Right. And to understand their hardiness, you really have to look at their ancestral home, which is the human gastrointestinal tract.
Your gut.
The gut. And the gut is a brutally hostile environment for a microbe. You have toxic bile acids constantly pouring in, intense osmotic stress, and wildly fluctuating oxygen levels.
Sounds awful.
It is. To survive that gauntlet, Enterococcus developed an extreme tolerance toolkit. For instance, it can grow comfortably in 6.5% sodium chloride.
Wait, 6.5% salt? That is incredibly high. If you put a normal bacterium in an environment that's salty, the osmotic pressure would just suck all the water out of the cell, and it would shrivel up like a microscopic raisin, wouldn't it?
Exactly. But Enterococcus pulls it off. It alters its cell wall and uses highly specialized molecular pumps to actively eject sodium ions.
Wow.
It's like it's constantly bailing water out of a sinking boat just to maintain its internal balance. Furthermore, it survives temperatures ranging from a chilly 10 degrees Celsius all the way up to a sweltering 45 degrees Celsius.
That's a massive range.
And it actively grows in the presence of bile salts. Clinical labs actually use a test called the bile-esculin test to identify it, precisely because it thrives in toxic bile where other bacteria just dissolve.
So because it evolved in this extreme toxic environment of the gut, it's essentially wearing like a heavy-duty microscopic spacesuit.
Yes. That's a great way to think about it.
And because of that spacesuit, being thrust out into the cold, dry world of a hospital ward is basically a vacation for it.
Exactly. That spacesuit is why it is so terrifying in a healthcare setting. Think about the last time you visited a hospital. The environment is aggressively cleaned.
Right, bleach everywhere.
Right. And most bacterial pathogens, if they land on a dry surface, they just dry out and die within hours. But Enterococcus uses that gut survival toolkit to persist on dry hospital surfaces. I'm talking bed rails, blood pressure cuffs, stethoscopes, doorknobs, for weeks to months.
Months on a doorknob, just waiting.
Just waiting. This is why it earned its spot as one of the IDS's escape pathogens, which is an acronym for the leading causes of hospital-acquired infections. It is a hallmark surface transmission organism.
So the risk doesn't leave when the patient leaves.
Exactly. If a patient colonized with certain resistant strains leaves a hospital room, that room remains a severe contamination risk long after they check out, because the bacteria are just sitting there in their spacesuits, completely unfazed by drying out.
Okay, so if this organism is built like a microscopic tank, and it can survive for months on a doctor's stethoscope, how do doctors kill it when it actually gets inside a vulnerable patient? Let's say it gets into the human bloodstream.
Well, this brings us to a really foundational concept in pharmacology, the difference between tolerance and killing.
You mean the difference between bacteriostatic, which just inhibits the growth of the bacteria, and bactericidal, which actively kills it dead?
Right. Enterococcus exhibits a phenomenon called antibiotic tolerance. If you treat it with standard cell wall active drugs, like, say, penicillin or ampicillin, it doesn't die.
It doesn't.
No, the drug just stops it from multiplying.
So it just hits the pause button. And I imagine for a lot of mild infections, pausing it is probably enough because your own immune system can sweep in and clean up the paused bacteria, right?
Exactly. But what happens in a severe infection, like enterococcal endocarditis, where the bacteria form a thick biofilm-like vegetation directly on a heart valve?
Oh, wow. In that case, the stakes are way higher. Blood is rushing past it constantly. The immune system probably has trouble penetrating that vegetation. You can't just hit pause there. You need bactericidal action to clear it out completely.
Precisely. So clinicians and researchers spent decades studying this tolerance, and they discovered a pharmacological solution based on synergy.
Synergy, okay.
You pair that cell wall drug, like penicillin, with a completely different class of antibiotic called an aminoglycoside. Drugs like gentamicin or streptomycin.
Okay, this sounds like medieval warfare to me.
How so?
Well, the penicillin is the battering ram. It hits the castle wall, and while it doesn't destroy the castle, it cracks the stone just enough.
Right.
And then the aminoglycosides, they are the assassins. They slip inside through those fresh cracks, bypass the main defenses, and target the king, which in this case is the bacterial ribosome. Neither the battering ram nor the assassins work alone, but together, it's game over.
That is exactly what happens. The aminoglycoside assassins normally just bounce right off the intact enterococcal cell wall. They cannot get in.
Right, because of the spacesuit.
Exactly. But when the penicillin battering ram damages the envelope, the aminoglycosides rush in, bind to the ribosome, and halt protein synthesis completely. Together, two static pausing therapies become one lethal bactericidal combination. It is just a masterclass in understanding the why of drug interactions.
That is so cool. So we figure out how to breach the walls and send in the assassins. But Enterococcus didn't just take that lying down, did it?
Not at all.
It evolved a terrifying new defense mechanism in response, which leads us to the emergence of vancomycin-resistant enterococci, or VRE, the famous superbug.
Yes, VRE was first reported between 1986 and 1988 in Europe, and it showed up in the United States shortly after that.
And the molecular mechanism here is just wild, because normally an antibiotic like vancomycin works by physically binding to a very specific target on the bacterial cell wall, right?
Right, a terminal peptide building block called diala-diala.
Yeah, diala. It locks on, comes up the wall construction, and the bacterium dies. But VRE carries these specific gene clusters, mainly called VAN A and VAN B, and those genes literally reprogram the cell wall construction.
They do. They change the target from diala-diala to diala-dilac. It alters just one single molecule in the building block.
But that one single change means vancomycin can no longer bind. It's like the bacteria secretly changed the locks on all the doors in the middle of the night, and suddenly the antibiotic's key doesn't fit anymore.
Now, what makes this an incredibly rich story isn't just how the bacteria changed the locks, but, why. What was the massive selection pressure that forced this evolution in the first place?
Right.
And this leads to what's often called the transatlantic natural experiment.
Because the timeline of how VRE spread in Europe versus the United States is totally different, and it all points back to agriculture, right?
Yes. In Europe, farmers widely used a glycopeptide antibiotic called avoparsin. And chemically, avoparcin is nearly identical to vancomycin.
Mm.
For decades, it was just mixed into livestock feed as a growth promoter. Animals ate it, and they grew faster and bigger.
But because avoparcin and vancomycin share the exact same mechanism, drenching European farms in avoparcin essentially trained the bacteria to resist vancomycin too.
Exactly. Agricultural avoparcin use drove massive cross-resistance in the enterococci circulating in farm animals. It created this enormous pool of resistance genes. And in Europe, VRE actually became widespread in the general community first, long before it started causing major outbreaks in hospitals.
Okay, I have to push back on this a little bit.
Sure.
Because human intensive care units and pig farms are completely different worlds, how does a bacteria from livestock feed actually make its way into a sterile hospital 5E line to cause a deadly infection? Are we really blaming farmers for an ICU problem?
I know it seems like a massive leap, but this perfectly illustrates the One Health framework.
One Health.
Yeah, the core of One Health is that agricultural ecosystems, the environment, and human health are all inextricably linked. You can't separate them.
So walk me through that transmission web. How does it happen?
Think about the runoff from those farms getting into the local water supply. Think about the farm workers handling the animals or their meat processing pipeline in general. VRE doesn't immediately cause disease in healthy people. It just quietly colonizes their gut.
Ah, I see.
So you have thousands of healthy people walking around the community with VRE in their intestines. Then someone gets into a car crash or needs major surgery. They go to the ICU. They get pumped full of broad-spectrum antibiotics that wipe out all their normal, susceptible gut flora, and boom.
The VRE, which is immune to those drugs, suddenly has no competition.
Right. It takes over, escapes the gut, and causes a catastrophic infection.
Oh, wow. So the hospital didn't create the superbug. The hospital just provided the environment for the superbug to reveal itself.
Exactly. And the proof of this farm-to-hospital pipeline is in the transatlantic contrast. The United States never approved avoparcin for agricultural use.
So American farms weren't a breeding ground for this specific resistance.
Right. In the U.S., VRE did not emerge in the community first. It emerged strictly as a hospital-acquired pathogen, driven entirely by the heavy use of vancomycin inside human intensive care units.
That is fascinating.
Two completely different ecosystems, two different evolutionary pressures, leading to the exact same terrifying result. The evidence connecting the farms to human infection control was actually so overwhelming that the European Union officially banned avoparcin as a feed additive in 1997.
That is just a staggering real-world consequence of biology. But because Enterococcus is so adaptable, even modern hospitals frequently misunderstand it, don't they?
They do. Let's start with infection control failures. We just talked about the Van A and Van B resistance genes, which cause these massive spreading VRE outbreaks.
Right.
Well, there are other less common species of Enterococcus, like Enterococcus gallinarum and Enterococcus castelliflovus.
And they have different resistance gene, right? Than C.
Yes. But here is the critical difference.
Yeah.
The Van C gene is intrinsic and chromosomal. It is permanently locked into their DNA. It cannot be transferred or shared with other bacteria.
Okay, whereas Van A and Van B are carried on mobile genetic elements.
Plasmids and transposons, yeah.
I always picture plasmids like little genetic USB flash drives. The bacteria can just copy their resistance files onto the flash drive and hand it to the bacterium next to them.
That's a great analogy.
But Van C doesn't have a flash drive. It's just permanently bolted to the hard drive.
A brilliant way to phrase it. But hospital labs will sometimes run a basic automated susceptibility test, see that a Van C organism is resistant to vancomycin, and just panic.
Because they think it's VRE.
Exactly. They mistake this non-transferable resistance for a highly contagious Van A or Van B outbreak.
So they throw the patient into strict isolation, force nurses to wear full contact precaution gowns, and burn through thousands of dollars in resources.
All for an organism that has absolutely zero potential to cause a sweeping ward outbreak. Treating a Van C isolate like a contact precautions emergency is a massive, documented overreaction. You really have to understand the genetics, not just read a basic drug test.
We also need to loop back to that paradox I brought up at the very start.
The probiotic one.
Yeah. How is it possible that the exact same species, Enterococcus faecium, is simultaneously a feared hospital superbug causing endocarditis and something I can buy right now as a probiotic supplement for gut health?
Well, certain strains of Enterococcus faecium, specifically one known strain SF68, are widely marketed as probiotics and used in animal feeds.
But how?
It forces you to rethink how bacteria work. Virulence, which is the ability to cause disease, is strain-specific, not species-wide.
Okay.
Think of it like dogs. A golden retriever and a timber wolf share a massive amount of DNA, but you'd only let one sleep at the foot of your bed.
So the probiotic strains are the golden retrievers of the Enterococcus world.
Exactly. They lack the specific virulence factors and the mobile resistance genes that make the clinical isolate so dangerous. Though, I should add, pumping high concentrations of any Enterococcus into the gut remains an area of really intense debate among microbiologists.
I can imagine. Well, before we wrap up, we need to clear up two major misconceptions for anyone listening who might be feeling a bit paranoid right now. First, VRE does not mean untreatable, right?
No, it doesn't. Vancomycin resistance is serious, obviously. But we still have drugs like linezol and daptomycin that remain highly active against most VRE strains.
Okay, good.
We monitor them closely for resistance, of course, but the medical community still has weapons in the arsenal.
And second, despite everything we've talked about with endocarditis and ICUs, Enterococcus is generally harmless unless it escapes its normal niche. It belongs in your gut.
Right.
It only becomes a nightmare if it gets somewhere it shouldn't be, like into the bloodstream through a catheter or onto a damaged heart valve.
Context is everything with this bug.
Absolutely. So if we synthesize everything we've covered, Enterococcus is truly a marvel of evolutionary survival. From its dramatic taxonomy split from Streptococcus, to its sheer reliance on extreme hardiness, to its starring role in the global antibiotic resistance debate, it absolutely demands our respect.
It is a survivor, first and foremost.
But I want to leave you, the listener, with one final provocative thought. We mentioned at the very beginning that Enterococcus is used by the EPA as the standard fecal indicator organism to test whether coastal marine waters are safe for swimming.
Right, and they use it precisely because it survives the extreme salt of the ocean so much better than E. coli. It shrugs off the ocean salt the exact same way it shrugs off the bile in your gut.
And remember those Van-A resistance genes we talked about? The genetic flash drives that reprogram the cell wall? Well, they are highly mobile. They are so mobile, they have even been documented transferring out of Enterococcus entirely, jumping into Staphylococcus aureus to create vancomycin-resistant Staph aureus, or VRSA.
It is a phenomenal example of horizontal gene transfer.
So here's the question for you to ponder. If this incredible, indestructible survivor is constantly flushing into our coastal waters, surviving the ocean's salt, and we know it carries highly mobile resistance genes shaped by decades of agricultural runoff and hospital wastewater.
Oh, wow.
Are our oceans quietly acting as a giant, invisible mixing bowl for the next generation of horizontal gene transfer? Let that mull over in your mind the next time you go for a swim at the beach.